So today I want to talk to you about systems neuroscience, which is loosely defined as the study of structure and function of intact neural circuits at all levels of analysis. And so I think this captures very well the the conundrum and complexity that today's neuroscientists are faced with in that to be able to make sense of what the brain is doing, one needs to be able to span, or one needs to be able to understand across different scales what is happening inside the brain. So starting from, cellular and molecular levels up to the level of cognition and behavior and the mind. So this is an extremely vast field, both in the physical sense, starting from molecules, going up to cognition, but it's also vast in the sense that of the techniques that are being used to probe neuronal function, etc. So. And indeed, the picture is not as nicely defined as I presented here, in that we now with modern techniques that are available to us, we have cases where we are able to span these dimensions. So we're able to record or image single neurons with this fine resolution that you see here, up to the level of the synaptic button. But for increasingly large populations, and I have here an example to show you, which is the case of zebrafish. So these are fish that are actually transparent, and these fish are optogenetically or genetically manipulated to transmit fluorescent signals or light every time a neuron spikes. So now with this type of techniques, and we see here, we're able to record, as in we is the neuroscience field, able to record from 80% of the brain from these small fish. So increasingly the techniques are getting better and better, and the ability to span these levels and to ask questions that span the levels becomes increasingly possible. So this is an imaging technique, and I will be primarily talking today about electrophysiology, which is a technique that's applicable from basically from cellular level up to the level of circuits, pathways inside the brain, looking at interactions between brain areas, but also these techniques are being used in humans, and maybe some of you have heard of recent advances in that, for example, Neuralink. Okay, so yeah, this is roughly so what is electrophysiology? We call it in vivo electrophysiology because we do it in living animals that are behaving. So it's a technique with which we can record the aggregate electrical activity inside the brains from inside the brain and from many cells in the vicinity of an electrode. And so the field is also advancing quite rapidly, I guess. So from the 70s, where we were able to record tens of neurons using very rudimentary wires that were spun together. So tens of neurons into the 90s, where we use essentially circuits, printed circuit boards that we put inside the brain. And on these circuit boards, we have many electrode contacts, and from that we can record across many positions inside the brain tissue. And into the 2010 or 2015 or so, with the introduction of CMOS-based microchip electrodes that actually have thousands of electrodes on them, which basically tile the whole surface of the probe that we put inside the brain. And using this, we're able to record up to thousands of neurons at a time. So it's an exciting field exciting time to be in the electrophysiology field, in my opinion. Um, and so what do the signals look like? I want to just give you the very basics here. You're looking at a recording here from the hippocampus of a mouse that I collected. This is using a silicon probe, and we have a bunch of 64 actually channels that we're looking at here, in the space of less than 500 or 600 micrometers. And there is essentially two things that we can extract from these types of signals. There's more, but I will only mention two. One is aggregate activity that stems from, basically a lot of neurons being active together, and sort of currents and ionic currents flowing inside and outside the neurons. And this these ionic flows give rise to oscillations that we see here. This particular oscillation is called the theta oscillation, which just by looking at this, I can tell that the animal now is running, because when the animal is running in the hippocampus, we see this oscillation. Okay, so um, further, we can look at, we can identify faster oscillations. This is an example here of a sharp wave ripple, which is an event inside the hippocampus. This is an event which lasts for about 100 or 200 milliseconds, maximum 200 milliseconds. And the oscillations that we observe here are of the order of 150 hertz or so. These are very important events in the brain. Maybe we can mention them later. So this is one type of information we can extract, which is the oscillations. And then there is one more critically very important one, which is the spiking of individual neurons from these recordings. So essentially every little spike that you see here is a recording of an action potential emitted by some neuron inside the brain. And by virtue of the way we arrange the electrodes, we are able to essentially see that this particular spike here is coming, there's multiple copies of it on across 4 or 5 channels here, and we can assign basically this event to a particular cluster. Perhaps this event here is from a different cluster than this event here, or maybe from the same one. But using complex computational methods, we're able to assign these different events to putatively to different neurons inside the brain. And from there on, we can ask questions about what is this single neuron coding for, or what is its activity reflecting in terms of the behavior of the animal. So using these techniques, we can record these two, or we can extract these two types of modalities. Now, I will start talking to you now about one of the studies that I'm actively now working on in terms of analysis. This is data I collected not long ago from the hippocampus of mice. And the hippocampus is, for those that don't know, is the main brain structure inside the brain that's associated with working memory and memory consolidation. So this is a structure that's one of the first to degenerate in Alzheimer's disease, and hence the cognitive deficits and memory deficits that we see in these patients. And so at the same time, I think it was, I'm pretty sure it was in the 70s that it was first shown that in the hippocampus of animals and later in humans as well, that single neurons tend to emit spikes at specific positions within the environment of the animal. So this is the hippocampus here of the mouse. We put our electrode down into this area here, which is the cell layer. And from there, we can record many neurons. And this is an example from the Wilson lab. Each color here represents spikes that were assigned to a particular neuron. And we see that, for example, the pink neuron here likes to fire only in this vicinity. The green one fires a bit more variably in this area or has a bigger field, and so on and so forth. with the important thing being that if you sample from enough of these neurons, you are able to tile the whole space that the animal has to navigate across. And so this gives rise to the hypothesis that this place cells form the neural basis for a cognitive map. So it's a map inside the animal's head, which basically encodes everything that the animal needs to know about its environment. Here's another example where it's a mouse that's behaving here in a two-dimensional arena. And in the same manner, we find cells as the animal covers the space many times. We can see cells that fire at specific positions in the environment. But things are not so simple because this is not a static map. So this is a map that tends to vary across time. And this has been known for a while. Okay, so so there's two ways essentially we can think of variability in this spatial map that these cells represent. So the first is that there is variability in the external world, which then needs to be reflected inside this inside the brain with this place cells. So if we take a rectangular arena and we let the animal roam inside, we can form a spatial representation with these place cells. But then in exactly the same space, we change the walls of the arena from a rectangle to a circle. Then what we see is that there is a remapping. So the same cell that was firing here with a very low rate now starts to fire at a different position. So this is perfectly fine and expected. It happens for most cells. In some cases, it doesn't happen. But this is variability that's, let's say, easy to explain, at least conceptually. But if we take an animal and we get it to, for example, this is in a linear track. This is actually a circular track where position here would be the 360 degrees of the circle. So when the animal is traversing this environment, there is this particular cell 349, that emits spikes only or with very high probability at this location here, 120 degrees. But if we look more closely, we see that, for example, in this trial, there was actually no spikes at all. In other cases, there was many spikes, and so on and so forth. So there is still variability, despite the fact that we haven't changed anything in the environment. And so the question I set out to investigate, or one of the questions with this setup that I built, is where does this variability come from? The one that cannot be attributed to the outside world. So I set up this experimental apparatus, which basically involved having a mouse head fixed, allowing us to put electrodes into its brain in and out on a daily basis. And the animal was allowed to run on this disc over here. And depending on how fast the animal was running, this virtual or semi-virtual environment in front of the animal would start to spin. And by spinning, it would bring about different objects. And therefore, what we would expect to see is that in the hippocampus, a spatial representation would be formed, or more simply, we would be able to record place cells tuned to particular positions in this environment. So the critical thing here is that together with electrophysiology, we were also recording through a series of cameras that you might be able to see here, and another one that's out of view. We were able to monitor what the animal is doing in terms of its facial expressions, its where it's orienting its attention, using its using its sensors like the nose, the whiskers, and etc. So by doing this, we were hoping to capture variability in this spatial representation that correlates or can be attributed to what the animal is doing and how the animal is sensing the world. So importantly, we opted for this type of setup, like a physical setup rather than a completely virtual one, because it also allowed us to put important stimuli on to have the external modification of the environment, such as conspecifics, for example. And I will show you now a quick video. This is an animal now that's waiting for this light to turn off. As soon as it turns off, he knows that he can now do his task. And so as he runs, the environment automatically through a motor spins. And so now he sees a novel stimulus for the first time, and he stops a bit to check out what's happening, but he continues to run and explore. Continues to run. In fact, he's running because he's thirsty and he wants to get some water rewards, which he gets after each time he goes around the environment. So, and during this process, as you see here, we have our silicon probes lowered inside the brain in the hippocampus, and we record brain activity at single neuron resolution. So here you see a close-up. These are the electrodes. This is electrodes penetrating inside the brain. There's actually 8 shanks across 2 probes here with 128 channels. And this is a typical recording session. So these are all the channels that we have, or almost all of them. And you see, in line with what I showed you before, that on each one of these shanks, signified by different colors, we have spiking events that I hope you can see, which we can then assign to particular clusters or to particular single neurons. Moreover, what we do is once we define these clusters, we can then divide the cells into excitatory cells based on the waveform of the spikes the average waveform of the spikes, or into inhibitory neurons based again on the spike width. And accordingly, these cells are known to have low firing rates and high firing rates. These are modulated by the theta oscillations in the hippocampus, which we see here in the interstimulus interspike interval, I'm sorry, with a peak at the frequency of theta, which is 7 Hertz. Whereas for the inhibitory cells, their interspike interval is not as well defined as here, and in fact, it happens across many different timescales. So having done that, we can then ask to what extent individual cells, as we've identified them here, are tuned to the position of this virtual carousel environment. And indeed, for different conditions, such as baseline, where we do no manipulation to the environment, or when we expose the animal to a conspecific or to a novel object, we see that in all these cases, cells have our place cells tile the whole environment reliably. I should mention that this is cells across 3 or 4 animals. So if we look at individual animals, then this coverage won't be as perfect as you see it here. So now we know that we have place cells. So now we can start to ask questions about individual cells or individual place cells and about their variability. These are cells, example cells from different recording sessions. So we see here marked with red. These are cells which more or less change their firing rate, or some do, some don't, when we introduce a manipulation in the external environment. So for example, this one fires more or less reliably until we introduce a novel object, and slowly after 20 trials, it stops firing. Here's another one where we have exposure to a conspecific, where the cell was firing in this position, and then it stops firing as soon as we introduce the change in the environment, and so on and so forth. We see all sorts of responses. In some cases, cells don't change at all. But also we have cases where there's no change to the environment, and as I already alluded to, in those cases, we still have variability. So here we saw almost no spikes in this position for this cell, whereas in other trials, there was quite a bit of firing. reinforcing the point that there is variability in the absence of external of changes to the environment. Okay, so as I mentioned before, we have this series of cameras, and what we do is we monitor along with a bunch of other variables that I don't really have time to mention. One of them is the speed, for example, of locomotion. Um, we monitor what the animal is doing with its face, essentially, which turns out we can read a lot from a mouse's face using these close proximity cameras. I'll play you a video now where you can see the variation in the pupil size. As you see now, it's getting smaller. Um, and the animal now is drinking. We can monitor the position of the pupil, and we know the position of the animal in this carousel space. And at the same time, we can look at the nose position, as you see here. Is the animal orienting its nose towards the direction that a new stimulus is coming from? Is it orienting its nose towards the past or towards the stimuli that already passed? We can monitor the pupil position, the ear position, and so on and so forth. So this is a very rich, multi-dimensional behavioral space that we can exploit to then ask questions about how this behavior reflects on the variability of individual place cells. So first of all, to point out that indeed the behavior is variable as one would expect, and there is a bit of tuning of this behavior according to the position of on this carousel space. So here's an example of the pupil size when we introduce a conspecific, and what we see is that as soon as we introduce the conspecific, there is a huge increase in the size of the pupil. And again, this is maybe not so surprising because we would expect to see a strong response to a social response, which could be expressed through the pupil size, which is sensitive to arousal level and novelty and all sorts of neurotransmitter changes inside the brain. Similarly, we can look at, again, the pupil size for baseline conditions. Indeed, here it looks much more stable, although still we see some variability. And simply what we need to do now is try to map this variability that we observe in this behavioral space onto the variability that we observe, as I showed you before, in the neural space. And so this is a little bit analysis heavy to explain to you in detail, but suffice to say that following a bunch of shuffle testing and conditioning the signals, etc., and z-scoring and what have you, we can show that essentially this graph shows you how activity of individual cells, which are the circles that we see here, is affected by this particular behavioral variable, which is the respiration frequency. So how fast the animal is breathing or sniffing the environment. And what we see for baseline conditions is that there's a bunch of cells that are affected by this behavioral variable. However, there's some that are centered around 0, which implies that they're not affected so much. And in fact, this is wrong. This should just say Z score instead of p-value. But we can also ask the same question in the case of when we change the environment through placement of a conspecific. And here we see a much greater, a stronger response or more cells responding, essentially, more cells changing their activity as a function of respiration frequency after this manipulation. So this is like a composite of behavior, cell activity, and position analysis. So this is ongoing analysis, but this gives you a flavor of what it is that one can do with this data. So we go from single neurons to asking questions about cognition using essentially mice that we can't really ask them what they're thinking, but using theoretical frameworks, we can reach some kind of conclusions. So, and this just as the last slide for this section of my talk. This speaks also to the way we think about processing in brains, and you have here the classic view, which is essentially the computer analogy, where you have sensory input coming into this passive device, which is the brain. Some data transformation takes place, and then this gives rise to a motor output. So this is the old way of thinking of things. Now this is giving way to a different way of thinking of brain processing, and this is centered around active sensing. So in order for the animal to form any kind of representation, or indeed to interact with the world, it needs to first it achieves this through active sensing. So if the animal doesn't move, if the animal doesn't try to do something, it won't be able to form any sort of representation. And this has also a lot of implications for robotics, artificial intelligence, etc. So the new way of thinking of what the brain is doing is as a device that interacts with the world uh, in order to generate a map or a representation of the space, and then use this representation to again act outwards and accordingly modify the environment. So with that, I will conclude the part of my talk where I talk about these little creatures, which have been extremely and are still extremely useful in neuroscience and in biology, in research in general. And I will move to bigger creatures. So this is work I did in Cambridge. And it concerned the development of sheep as a large animal species for systems neuroscience investigations. So a quick motivation of why we would want to bother with using yet another species. We have a bunch of them here. And if we look at in vivo experimentation, because we have other types of experimentation, either in the dish or on a computer. If we look at in vivo experimentation, we have a bunch of species that we can study or we can select from, starting from the nematode, which has about 1,000 neurons, the zebrafish that I already talked about, which has, I think, 100,000 neurons, and moving up to, up to primates. So obviously there's trade-offs. If one experiments with small animals, you can make nice inferences about the organism as a whole. You can scale your research very easily. You can record from, tens of these at a time. But what happens is that you lose your ability to translate, as if we are comparing to larger animals, for example, primates. The behavior is quite different. The brain is quite different. So there is trade-offs, scalability, translation, and the scale at which we can monitor brain activity. So the question here is, can we find a species? And now you already know that this species is going to be sheep, that sort of fills this gap between rodents and primates. And I should also mention that another thing that is of concern is, as you move towards this direction to the right of the graph, the ethical concerns also increase, and so does the cost of experimentation. So sheep seem to be a good species that fits inside this this area here, with increased ethical, decreased ethical concerns when compared to primates, but increased translatability when compared to rodents, and many other benefits. So to start this program, we began with EEG work. So I developed the methods to record essentially from freely moving sheep using a series of intracortical screws implanted on both hemispheres of the cortex of this animal, and using various techniques such as ambulatory amplifiers or wireless amplifiers, we were able to do some chronic monitoring of brain activity in order to track disease progression. So one area that I'm quite interested in, which is sleep, features very heavily in neurodegenerative conditions. So the question here was whether we're able to use EEG as a biomarker for neurodegeneration in some sheep that carry naturally occurring diseases. So this first disease was Batten's disease. It's a lysosomal storage disorder that afflicts children, which unfortunately die at a very young age. The symptoms of this condition is the loss of vision, seizures, dysphagia, severe degeneration, and sleep abnormalities. So the cortical loss and the loss of vision were known in this particular natural occurring variant of Batten's disease in sheep. And so we set out to use the EEG technique to show these other deficits, which indeed we show, and I will just fly through this a bit quickly. So in terms of sleep, perhaps unsurprisingly, we showed that when the animals sleep and the cortex enters this slow wave deep sleep mode, the sleep was or the amplitude of these slow waves was much smaller in the affected sheep compared to the age-matched and age-matched unaffected sheep. But as I said, this is not a big surprise because although this is end stage, but you can very well see that the neurodegeneration is massive. Now, if we look at the occurrence of these slow wave events, and we use our knowledge of high frequency activity and how it locks onto these slow wave events in the brains of humans and mice and now sheep, we see that for the healthy sheep, this is easily shown. So this 18 hertz activity is phase locked to this particular phase of the slow wave oscillation. But if we look in the affected sheep, this is not the case. And this is perhaps a little bit more difficult to explain just by sheer um, volume conduction deficits by the shrinking of the tissue. So moreover, just to reinforce the sleep abnormalities, um, we did here a sleep deprivation study, which is known after sleep deprivation. We know exactly what to expect in terms of rebound of sleep, in that our sleep after a sleep deprivation event is going to be of longer duration. And we expect to observe higher amplitudes sleep taking place at the early stage of the sleep episode versus the late stage of the sleep episode. And this should be more pronounced after sleep deprivation. Indeed, in the affected sheep and also in the healthy sheep, we see that the way the amount of sleep does increase after sleep deprivation, dramatically. So maybe more dramatically in the unaffected sheep than the affected, but it increases in both. But at the same time, if we look at this profile, that we expect to see a decreasing amplitude of slow waves, which implies that a reduction in depth of the sleep, a reduction in sleep deficit, essentially. This was not observable in the Batten's disease sheep. So perhaps because of the degeneration, the sheep are not able to recruit enough or recruit coherent slow wave activity enough so that they diminish or reduce this sleep deficit within a single sleep episode or a single night. Um, Now, I did mention that Parkinson's disease patients suffer from seizures, and indeed we showed this also in the Parkinson's disease sheep. This was extremely obvious and inexistent in the unaffected sheep. I won't spend too much time on this. Um, Mostly this was taking place during wake periods, but it also happened in periods of sleep. So finally, just the last thing about EEG, in the same animal cohort, the Batten's disease sheep, um, and since we know that patients have dysphagia symptoms, we looked at rumination as a motor essentially output. And rumination is this thing in sheep where they regurgitate their food in order to masticate it again and break it down a bit more before they swallow it. And when sheep do this activity, it has a very, very fine structure, temporal structure to it, at the level of rumination episodes that last 50 minutes, and rumination bouts whereby the sheep here swallows, then regurgitates, and then starts to masticate, and then this repeats all the way down to the chewing intervals. And for all these time scales, we observe that in the affected Batten's disease sheep, the rhythmicity is very well or very much disrupted for all of these sheep. Um, so um, so the interim conclusion here is that using intracranial EEGs, we can basically do longitudinal brain function monitoring in health and disease, and this method can be extremely useful in cases where we want to look at interventions such as viral vector gene therapies that we want to monitor over a long time to see any effect of the treatments. And so we also demonstrate here the translational nature of our findings. And finally, the last part of my talk is to look at how, looping back to the single neuron stuff I was talking about before with mice, whether we can do this with sheep. So this was quite a complex task to accomplish, mainly because no one had ever tried this before. But I can give you the spoiler now that this is indeed possible. So we made this implants here that you see. This is a bunch of electrodes that we can lower individually into the brain and creep up on individual neurons. These are called tetrode electrodes. And just because each one of these electrodes has 4 contact points, we are able to triangulate or parse different spikes to different neuronal clusters because we have different copies of the same cells on each electrode. And so we do this quite reliably in sheep now, in freely moving sheep. And as I showed you before, we can cluster them, we can divide them into principal cells that are theta modulated in the hippocampus, or interneurons that are less modulated by the theta oscillations. These are the implants. They reside inside these chambers here, and during the behavior, the animal is able to freely move while we record this single neuron resolution activity. Now, just to quickly show you that these animals, in fact, are quite happy to participate in these experiments. You see here one of the sheep that's willingly coming to do its testing, mainly because it knows it will receive reward, and it knows it's going to play around a bit with some stimuli, and they actually are very keen to participate in these experiments. And we observe basically no behavioral side effects from these procedures. And this is the type of experiments we see. I think you cannot hear the sounds, but just now the animal chose the correct symbol, and some reward in the form of pellets has dropped into this container here. The animal consumed the reward and is now approaching the choice point, and here it chose the wrong choice. and it gets no rewards, so then it knows it has to go to the other side to get some reward now again, and again it chooses the wrong choice, and so on and so forth. And throughout this behavior, we're able to record those single neurons that this is the correct choice here that I was referring to before. So now let's look at a signal here from the hippocampus. What we see here is the oscillation. And on top of it, live, we can extract the spikes that you see here. And let's observe first that as the animal starts to move, it consumes its reward now out of view, and now it's turning back to go to the other side. And as it starts to locomote, we see this very strong, very well-defined theta oscillation in the hippocampus, which actually groups the spiking activity of these neurons that are being detected from the same electrodes. And again, this is a stable recording. And you will see again that when the animal it consumes its reward, it's going to start to go out again, and we can again see the same theta oscillation appearing. Okay, so this is the technique, and I want to show you again just to convince you that cellular activity or activity of single cells is quite stable. Here, this animal has been performing two-choice discrimination for a few days, and it reached essentially 100% performance, as you see
Nikolas,
here.
sorry to interrupt you. Can we just, because we want to leave some time for questions, can we
Yep.
Wrap it up in about 5 minutes, if that's okay?
5 minutes, yeah, of course, yeah. So when we take these cells here and we center them around the choice points, we see that some of them fire more before the choice, some stop firing during choice, some do not really care about what the animal is doing and they just fire indiscriminately. But the main point here is that these cells are quite stable as the animal jumps around and behaves. And we can record a bunch of electrophysiological signatures. Maybe I don't have much time to go through this, but suffice to say that these are really translational signatures, particularly alpha oscillations that we don't really see in rodents, but we see in sheep. And again, to loop back to the results that I was showing you with the mice, we did record from the hippocampus, and we see cells, place cells that are tuned to particular positions in the environment, such as, for example, this one that only fires in this position when the animal is going in this direction, and not when the animal is going in the other direction. So despite the low number of cells we were able to record from the hippocampus, indeed, we were able to find cells that responded to place. And we talked before about remapping and the influence of cues, environmental cues, on the activity of these cells. Here's one more cell here that likes to fire only at this position, and only when the animal is approaching in one direction and not when it's going in the other direction. So what we did is we put a barrier up to obscure the experimenter here, who is a strong social cue essentially. And we were able to manipulate the firing rate of this cell quite easily. And at the same time, if we or after this, if we lower the barrier, we can reinstate the firing rate to the level of what it was before, as in it went from 6 hertz to 1 hertz back to 5.6 hertz. So I think this is, I think this is all the results I have, and I hope that this kind of convinces you that experimentation with large animals, such as sheep, offer really novel opportunities, especially because we're able to combine different techniques from imaging of the brain and physiology and large-scale behavior across, naturalistic environments. This is quite, original work and work that hopefully we will be able to continue in the future. But to focus back on mice and just to tie this with what I aspire to do in at the University of Nicosia, just with very quick observations that the neuroscience research in Cyprus is still at a growth stage. There's, in fact, very few players in this field or stakeholders, and I think this is a really good opportunity to do some synergistic efforts to be able to span all those levels that I showed you in the first slides. And essentially, I would like to argue that the University of Nicosia, the medical school, is in a really good position to lead efforts for systems neuroscience research and really to bridge the gap between the cellular work that takes place at the Cyprus Institute of Neurology and the human work that takes place at Sing and other places such as University of Cyprus, behavioral work at the science, and also some companies getting involved in this field now as well. So yeah, I think that's all I have to say. Let me just thank the labs that I worked in. The lab of I got the names wrong here. So this is Jenny Morton at the University of Cambridge, and this is Professor Anton Sirota at the University of Munich, and all their lab members, of course. And